So in our previous video, we learned quite a lot about the history of how DNA was first discovered and determined to be the heritable material that's found inside of cells. We also learned a little bit about DNA's structure and its properties. And so in this video, we're going to continue on our adventures by learning a bit more about how DNA replicates in prokaryotes and eukaryotes. So this is going to bring us back to a picture I talked about a little while ago where we examined the processes of replication and transcription, and these processes, as a reminder, can't occur at the same time. So this process of replication takes place during the S phase of the eukaryotic cell cycle. So that's where we are at the moment, is in the middle of that S phase. So a useful thing is that the mechanisms for replicating DNA are very similar across a wide range of organisms. And in particular, what that means is that we can go back to a simple prokaryote, E. coli, as a model system for studying replication. And the biggest difference between prokaryotes and eukaryotes, again, is how DNA is organized in the cells. So while the process is the same, there are some slight differences between them. So in a prokaryotic cell, remember, DNA is circular. There's just one single big circular chromosome. And so what that means is there's a single point where replication starts. That's referred to as the origin of replication, as you can see in the slide here. And from that point, basically the two strands of DNA get pulled apart, and then replication proceeds away from that starting point in both directions. So as it gets underway, there are two what are called replication forks that start traveling in opposite directions away from each other. And so those are illustrated here. And then eventually they reach the final part of that circular chromosome, and the end product will be two new circular chromosomes. And in those two new circular chromosomes, since replication is a semi-conservative process, one of the strands will be from the original double-stranded molecule, and the other strand will be a brand new strand that just got synthesized during the process of replication. We're going to be working with a lot of cartoons of this process, but I also think it's incredibly valuable to see real pictures of this process. So there's a couple of real pictures on this slide as well. If you can't see it completely because my head's blocking it, check the lecture slides in addition. But here in the lower right, we've got a picture of this process of replication taking place in an E. coli chromosome. So this is a scanning electron microscope image. And you can see a circular strand of DNA, and there's two little red arrows here that are pointing to those replication forks. So that in this case, replication is going to continue kind of around and down to replicate that piece of DNA. So the process is similar in eukaryotes, but with a small exception. So again, eukaryotic chromosomes are built of a single long piece of DNA, and I now do remember the fun fact about how long the DNA is that's inside of a single cell. It's about 6 feet long in a human cell. But there's a bunch of linear chromosomes instead of a single circular chromosome. So in a eukaryotic cell, there can be multiple replication origins on a single piece of DNA along a single chromosome. So that's illustrated in this other SEM image that you see. So each of these red arrows is pointing at a replication bubble, and on either end of that bubble, there's going to be a replication fork where replication is going to proceed away from the middle of that bubble. So the process of replication has a lot of players and different pieces involved, and I think it's really helpful to kind of study a range of depictions of this process. And in addition to that, I just found some really cool footage of replicating DNA from a paper that came out just 3 years ago in 2017. So prior to this point, we had no method for actually visualizing how DNA replicates, but in this paper, they were able to figure out a process so we can actually visualize it. So let's look at that footage for a moment. So I feel like I should introduce this footage with my best nature documentary voice here. So here we are looking at some DNA, and you can't see it now, but what's happened here is this DNA has been, or has had a dye added, a fluorescent dye that only binds when there are two strands of DNA together. So it's going to highlight places where replication is happening. So let's watch what this video shows. We may loop it a couple of times. You can see it's sped up. Okay, let's watch that again. So one thing to notice that's really cool here is we used to think this happened as a very smooth, continuous process, but this video shows us that actually it occurs in fits and starts. So the other thing we learned from this is we used to think that the leading strand and the lagging strand, I'll talk more about those in a minute, that those are replicated more or less simultaneously, but being able to actually visualize the process showed us that no, that's not the case. They actually can be replicated out of sync with each other at certain points. So you know, in watching this, I think it just emphasizes that for all that we think we know about how these processes work, there's still a ton of stuff that remains to be discovered. One more time for good measure here. Okay, so that's DNA live action getting replicated. And there's a couple of other links here that touch on some other concepts associated with DNA replication. I'm not going to go through them right this moment. We'll go through them during our lecture time. But the links are here in case you want to go through and watch them again. Instead, what we're going to do here is we're going to do a DNA replication draw along. This is very much like mitosis and meiosis. It's a case where I think that actually drawing out the process is a fantastic way to learn the process and know the process well. And in addition to that, you're going to discover that I may ask you questions that show some pictures of the process of replication, so it can be really useful to see how I draw this process out so that you'll be able to interpret those pictures more easily in the future. Okay, so let's get right to it. Okay, so let's just start out by getting ourselves oriented to a piece of DNA. And in this case, I'm not going to draw out the molecular structure in a lot of detail. You can go back to one of the biochemistry videos for that. Instead, what I'm going to do is I'm just going to draw it symbolically. So what I've shown here, this is going to represent the carbon phosphate backbone. And then at every position where there is one of those four nucleotide bases, I'm just going to draw a little tick mark to represent each of those bases. Right, so each of these represents one of the single strands of DNA, and as a reminder from that biochemistry video, these strands are directional. So at one end there's going to be a loose 3' carbon, so we refer to that end as a 3' end, and then at the other end there's going to be a loose 5' carbon, so we'll refer to that as the 5' end. And these two strands run anti-parallel, so they run in opposite directions from each other. So what that means is if up here is the 5' end, then opposite it there's going to be a loose 3' end. And vice versa at the other end of the second DNA molecule. So these are complementary to each other. So I'm just going to fill in the rest of the spots for these bases. Okay, and now we're ready to get underway with DNA replication. So the first step in the process of DNA replication is the process of unzipping the DNA. And there's a whole bunch of proteins that are involved in this process. So the protein that does this unzipping is a protein that is called helicase. And I like to remember it, well, first off, we can remember all these things are proteins because they have the suffix ase on them. So that tells us the thing we're dealing with is a type of protein. And helicase, actually, as it's unzipping, it spins around. So I like to think of it as being kind of like a little helicopter spinning around and unzipping our strands of DNA. So I'm going to just redraw this piece of DNA unzipped. I'm going to wind up with a replication bubble. Okay, and then I'll draw on my helicase here in blue, unzipping. And there is actually going to be two helicases here, one at either end. Okay. So now what that's done is it has exposed each of these individual single strands of DNA. But now I hope you remember that the carbon phosphate backbone on a piece of DNA is polar, so it's going to interact nicely with water. But these nucleic acid bases, nitrogenous bases, are nonpolar. So they're naturally going to gravitate back towards each other, and they could easily re-anneal is the phrase that's used, or reattach to each other. So the next thing that has to happen is just keeping those apart from each other. And that's accomplished by a set of proteins called the single-stranded binding protein. So I'm just going to abbreviate up here SSBP. Keep the DNA apart. And we can draw those on this picture just with some green circles here. I'm just going to depict that towards the outside. And you have to just imagine that the single-stranded binding proteins are going to bind all the way along the outside of these strands of DNA to help keep the single strands of DNA apart from each other. So once that has happened, the next thing that takes place is the addition of some short pieces of RNA that are complementary to the DNA. And these pieces of RNA are added by an enzyme called primase. So primase adds short pieces of RNA, okay? And we'll get to why RNA gets added first in a moment. So that's going to happen, let's say we can picture that happening up here. Maybe it'll happen down here. And maybe it'll happen over here, okay? And maybe up here. So these short pieces of RNA, they're going to have a sequence that is complementary to the sequence of the DNA. So if, for example, we had a sequence here that is A, T, A, then the corresponding RNA primer is going to have a sequence of U, A, U up here. Yeah, that's not super critical at the moment. We'll come back to that point, but kind of useful just to keep in mind sort of how those pieces of primer match up with the DNA. So once we have primer on, the next step is going to be to start adding bases, DNA bases onto this chain. So we're going to have the addition of DNA nucleotides by an enzyme that's called DNA polymerase 3. And it might seem a little strange to start out with a DNA polymerase 3. They were actually numbered in the order in which they were discovered. So this is the third one that was discovered. And DNA polymerase 3 has sort of an interesting characteristic, actually shared by polymerases in general, in that it can only add bases in the 5' to 3' direction. So this is a one-way enzyme. It cannot build in both directions, just one direction. So the next thing we need to do up here is just analyze where can DNA polymerase III start to add nucleotides onto these RNA primers that have been attached by primase. So we need to evaluate where our loose ends are. So if we look down here at the bottom, this original strand of DNA says parent strand of DNA, here's the loose 5' end. So what that means is that the loose end that's hanging out up here is a free 3' end. And its opposite end is a 5' end. So if DNA polymerase can only add in the 5' to 3' direction, what that means is we can build in this direction to the left here. And in the same fashion, this next piece here, if you notice, here's our free 3' end. So up top we've got 5', here's our 3', and so we can only build from the right towards the left. And then what we'll find is up top is going to be kind of the opposite thing, right? So here's our 5' end on the parent strand. So up here we've got a 3' end, and here's the 5' end. Same thing for this other primer. So what that means is we can build to the right. So if I just draw on my DNA polymerase III here in red, chugging along. So DNA polymerase can get to work here, it can get to work here, it could get to work there, and it could get to work here. And so in all those places, it can start to add onto this RNA primer, add bases. And now here's where you might start to notice something. Okay, so down here, DNA polymerase is going to add bases until it bumps into this next piece of primer, and then it's actually going to stop and fall off. And in contrast, up top, once this DNA polymerase III gets started, as helicase continues to unzip, what that means is this DNA polymerase can kind of, like a train on the tracks, it can just keep chugging along, continuously adding. So this upper portion at this replication fork up here is that whole section that can be replicated continuously. That's referred to as the leading strand. So that's continuously replicated. And in contrast, we kind of have a problem down here where we can't add any more bases onto this primer in the opposite direction, right? Because this is a one-way enzyme. It can only build in the 5' to 3' direction, right? So we're fine down here on the lower left. We can keep building. And so on this left replication fork, here's our leading strand, right? But so we're going to run into the same issue up here. If we don't have any primer, we have no way to build up top on this left replication fork. So this lower segment is referred to as the lagging strand, and it's built in pieces. And those pieces are referred to as Okazaki fragments. After the Japanese biologist who discovered them. So what's going to happen up here is we're going to have primase come in again, attach another primer, and then DNA polymerase will hop on, it will chug along, and it will build up until it encounters the next piece of primer, and then it will fall off again. So this lower portion from this replication fork headed towards the right, that is a lagging strand. And the same thing up here. So for the replication fork that's headed to the left, we've got a lagging strand up here as well. Okay, so helicase is going to keep going and zipping. We're going to have continuous elongation up here. We're going to have pieces of DNA added in chunks down below on this lagging strand. And so, that can continue for quite a while. But eventually what needs to happen is we need to replace these pieces of RNA with DNA. So we have a final DNA molecule. So our fifth step in this process is going to be the replacement of the RNA primer with DNA. And that's accomplished by another DNA polymerase. This is done by DNA polymerase 2. Okay, so DNA polymerase 2 is going to come in, and so I'm going to erase my DNA polymerase 3 here. So DNA polymerase 2, let's draw that in green at each of these sites. So this DNA polymerase 2 is going to remove the RNA, and it's going to replace that with DNA. Okay. Okay. But there's still a small remaining problem, which is that there are still gaps between where that previous piece of DNA was getting synthesized and where the DNA got replaced by RNA. So the next thing that has to happen is sealing of those gaps. So there's yet another enzyme that specializes on sealing the gaps. That enzyme is called ligase. Okay. We'll draw ligase in blue. Okay, so our main gaps at the moment are here and here. So there we go. Ligase has sealed up our gaps. All right, that's most of what I want to show you on this picture. And now I want to talk about a couple of other things that are happening simultaneously with the process at the replication fork. So one thing we have to keep in mind is that DNA forms a double helix, right? So it's spiral. So you can kind of think of it as being like if you've ever played with yarn or rope that has multiple plies that are twisted around each other, the same kind of thing is true of a DNA molecule. It's kind of like something that has plies twisted around each other. So if helicase comes in and starts to unwind those pieces of DNA, what it actually does is it forces some of the coiling out to the left and to the right. And so it's going to cause, so here's my helicase unzipping. So it's going to actually cause what's called supercoiling away along the pieces of DNA that are further out from the replication fork. So there's another enzyme that is involved in this process that prevents that extra coiling happening upstream of replication. And so that is done by an enzyme that's called topoisomerase. So you notice that topoisomerase has the word isomer in it. And so what topoisomerase does is, let's say it's acting right up here, it actually will cut a little nick in one of the pieces of DNA, sometimes in both of them, and it will let it unwind, and then it will rejoin the DNA. So topoisomerase is going to nick the DNA to prevent what's called supercoiling. Okay. All right, we aren't quite finished yet, so if your brain's feeling pretty full, I suggest that at this point you should pause the video and then take a quick breather and then come back for 2 additional topics here. If you are ready, the next thing I want to talk about is mistakes. So DNA polymerase III is actually a really accurate enzyme. It's fantastic at matching up bases with the original DNA strand. So up here I've drawn my template strand in black and then my newly synthesized strand in blue. But DNA polymerase does make mistakes on occasion. And so it makes mistakes at a rate of 1 mistake per 100,000 base pairs. But if you look at the final product after a round of DNA replication, the error rate is actually a lot lower. So the final error rate is more like 1 in 10 million. Or sorry, not 10 million, 10 billion. Another 3 orders of magnitude. So it's 100,000 times lower than the baseline error rate of DNA polymerase III. And so how is it that we wind up getting such a low error rate compared to what that error rate could look like? Well, the first thing is DNA polymerase III can catch a lot of its own mistakes. So it actually will slow down. If it mismatches, then it will stop, and then there will be a pause. It will relocate the correct match, and then it will keep going. But in other cases, there's some DNA repair enzymes that can also help to fix mistakes. And so here is the team that's involved in that. So first, there's an enzyme called exonuclease. So like the name suggests, exonuclease, it comes through and it chops out the offending mistake. So let's say if I show it in green here, it's going to just chomp out this offending mistake. Okay. Then after that's gone, then there is another DNA polymerase that can come in and put in a replacement base that is DNA polymerase I will come in and will put in a replacement base. Okay. And then, actually I misdrew this, so it'll stick in a new base. And then last but not least, ligase will come in and will close up the gaps for that new base. Okay, so that's one of the challenges in replication, is fixing mistakes. So here's our DNA repair team. The other challenge has to do with what happens at the tips of chromosomes. So at the tip of a chromosome, for a linear chromosome, the tip is called the telomere. And the place where we run into problems is, so DNA can only be synthesized, right, in the 5' to 3' direction. So let's say that up here is one tip, and this is our 3' end. Down here is a 5' end, so we can synthesize heading off to the left. So, if we're starting the process of replication, primase can attach an RNA primer here, right? And then that's going to let DNA polymerase get hooked on, and then DNA polymerase can just chug along this lower strand, right? And then after that happens, it's possible for the DNA polymerase to remove this RNA primer, but there's nothing for the DNA polymerase to hook onto to build a little stretch to fill this gap here. No way that it can do that. So what this would mean is if our round of replication ended here, we have these mismatched ends here. The next time these strands go through replication, the upper strand is probably going to be fine. There will be the same capability as we just saw here. But for this lower strand, now the new strand that's going to be built off of it is going to have the same type of problem. So there's going to be a little gap here at the tip. And so each time this molecule goes through replication, the ends would wind up shortening. So pretty soon that's going to start chewing through DNA or through genes on this chromosome. That's a problem. So there is one more enzyme that can add bases to these tips. And it can't add complementary bases. It just mostly adds A's and T's. And so that enzyme is called telomerase. So it adds random A's and T's to the tip to prevent shortening. Okay. I hope you enjoyed the draw-along version of this process, so let's recap one more time using pictures from your textbook. So here's the early stages of DNA replication getting started. So we've got helicase illustrated here, unzipping the DNA. This cartoon also shows you topoisomerase cutting a little nick in the DNA to help keep it from supercoiling. And just to get us oriented, here's our 5' end of one strand of DNA and its 3' end. And down below we've got the 3' end and the 5' end. So as helicase is unzipping, there's also these single-stranded binding proteins helping to keep the pieces of DNA from reattaching to each other, re-annealing. And then we've also got primase here, which is adding a short RNA primer to help DNA polymerase get going. Just to reiterate, again, DNA polymerase can only add nucleotides in the 5' to 3' direction. And actually, the nucleotides that DNA polymerase adds actually come with a triphosphate group on them. That might remind you of something, perhaps, maybe adenosine triphosphate, our major source of energy transfer in cells. And in a similar fashion, this triphosphate group on this nucleotide provides energy to facilitate bonding and attachment to the new strand of DNA. So 2 of those phosphate groups are going to drop off. DNA polymerase is then going to catalyze the reaction that attaches this new nucleotide onto the growing DNA strands. So this process will then keep going in a downward direction here. So here's another illustration of the process of elongation of DNA off of that initial primer segment. So this is showing you the full replication bubble that contains 2 different replication forks. And in each of those forks, there's going to be a leading strand that can be synthesized continuously and a lagging strand that's synthesized in pieces, those Okazaki fragments. So again, that protein that works on elongating that piece of DNA is DNA polymerase 3, building in that 5' to 3' direction. And that will be a continuous process along that leading strand. Okay, looking again at the lagging strand replication, so same process with primase, attaching an RNA primer, and then that allows DNA polymerase 3 to make an Okazaki fragment in the 5' to 3' direction. Then that DNA polymerase 3 will detach, and we'll keep going, we'll keep synthesizing more Okazaki fragments. And meanwhile, DNA polymerase 1 will replace that initial RNA primer with DNA. And then as the last step for the Okazaki fragment, DNA ligase will form a bond between those DNA fragments. Now, here's a picture that might blow your mind a little bit, because as it turns out, in a lot of cases, that replication fork isn't really a fork. And instead, as best we understand it, the way that this whole system is put together, there's actually a helicase molecule that has two DNA polymerase molecules attached to it with these connecting proteins. And so as helicase travels along and unzips DNA, the leading strand gets synthesized continuously, and there's a funny process that happens here with the lagging strand that causes a piece of DNA, that single-stranded DNA, to run out away from the helicase, and then this DNA polymerase 3 attaches past a piece of primer and starts to add bases along one of the Okazaki fragments. I don't expect you to be able to interpret these trombone model pictures, but I think they're cool just for showing how this might mechanically work in an actual cell. This is just a restatement of what I described for proofreading repair, so although mistakes are really rare, they do happen. Just as a reminder, DNA polymerase 3 will catch a lot of its own mistakes, and if it doesn't, in a lot of cases, an exonuclease plus DNA polymerase 1 plus ligase will correct those mismatches to bring the error rate down to only 1 in every 10 billion bases. One thing I didn't note at that point is that when those mistakes are passed along, that constitutes a new mutation. And most of the time when this happens, there aren't any major impacts of those mutations. Either the mutation has no effect, or in some cases that mutation may be harmful. And we're going to see why that happens more specifically in the next week. But those mutations are also the source of variation that forms the raw material for natural selection to act on. And we'll also be talking about that later on in the semester. I also showed you what happens at the tips of chromosomes with the enzyme telomerase, which adds somewhat random stretches of DNA to the ends to allow, or to keep those ends from shortening. And that's it for DNA replication. So from here, now that we've talked about DNA replication, the next topic we're going to dive into is the topic of gene expression. So that's what's pictured here on the right-hand side. So we're going to break that process up into two major steps. First, gene transcription and gene modification, and then the second step, gene translation.